Brendan Dassey trial transcript, day 3, continuing on page 37, Special Prosecutor Gahn's questioning of Sherry Culhane. So you can make a determination whether someone may be the source of a biological substance at a crime scene? Yes. And is this DNA technology that you've just talked about used in other fields besides law enforcement and the forensic setting? Yes. Could you explain just some of those for the jurors? The technology, the PCR technology that this system is based on, is used in the medical community quite frequently. It's used for a lot of diagnostic testing. It's used to identify individuals from mass disasters. I believe most servicemen now give a DNA sample that is kept on file. So there are a lot of other applications. This type of technology is used in many other applications besides forensics. Now in this case here, the case that we're trying today, did you receive items of evidence from law enforcement agencies to perform DNA testing? Yes, I did. And when law enforcement submits items to the crime lab, do you generate some type of case file? Yes, I do. And could you just explain to the jurors how you'll go about generating your case file? Anytime evidence is brought into the laboratory, we have individuals referred to as evidence specialists. They take the evidence from the submitter, the police officer or agency, and we have a computerized barcoding system in the laboratory that keeps track of all of this evidence. So anytime a piece of evidence comes into the lab, we give it a yellow sticker with a barcode on it that is a designation of what the lab number is. So we give it a numerical value. Sorry, a numerical number. Also, each item of evidence is also given an item designation. So for instance, your number of the case, and then you'll have items A, B, C, D, E. So all of that information is given to the items when they come into the laboratory. It's put into our computer system. All of that evidence is put into storage, into our evidence storage, and remains there until the analyst needs it to actually examine the evidence. All of the documentation that we generate for each case is kept in a case file. So all of our information with chain of custody, who took it in, what happened to it, plugs all of the notes and all of the data that we generate from our analysis is kept in a case jacket. And that's the case file that's generated for each case. And did you bring your case file with you today? Yes. And do you need that to testify today? Yes. And your file, your case jacket, will that indicate what items of evidence you received and on what date? Yes, it will. And will it also contain the dates that you performed your analysis? Yes. I'm going to ask Mr. Kratz if he would bring you a number of photographs, which would like you first just to look at, and then I'll have a question for you when you've completed looking at those. Yes. Those photographs, do you know who took those photographs? I believe they were taken in our laboratory, and those are photographs of Teresa Halbach's RAV4, correct? That's correct. And that was brought to your crime laboratory from the Avery salvage yard, is that correct? Yes. Okay, and who is Ron Groffey? He's one of our imaging analysts, and he takes the photographs when we process vehicles or when we need evidence photographed. And did he take these photographs? Yes. And as you look at each of those photographs, do they appear to accurately depict Teresa Halbach's RAV4 as it appeared when you first saw it? Yes, I do. Okay, I would like you first to look at Exhibit 141, and yes. Would you, and is it the photograph that you have in your hand, is that the same photograph that we have up on the screen? Yes, it is. Okay, and would you explain what that is? This is a photograph that it's taken in our garage at the laboratory, and it is a picture of the Teresa Halbach's RAV4 as it was in our laboratory. And when did you first see Teresa Halbach's RAV4 in your lab? November 7, 2005. And that would have been on Monday? Yes. Okay, and what was your involvement at this point on November 7? I was asked to process the car for the presence of blood or any biological materials. And how did you go about doing that? The first thing we do anytime we're processing evidence, whether it's a vehicle or a piece of clothing, the first thing we do is a visual examination, and we basically just look at the item of evidence to see if there's any obvious stains. We're looking for different biological materials depending on what type of case it is and what circumstances there were. And did you find any blood stains in Teresa Halbach's RAV4? Yes, as I was processing the car, again, I was just basically looking at the interior, and there were numerous obvious stains that were consistent with the appearance of blood stains. And did you perform any type of preliminary tests on these, what appear to be blood stains in Teresa Halbach's car? Yes, I did. And what preliminary tests would those be? Preliminary tests in the laboratory are tests that are not specific for a biological material, but it gives us a good idea that what we're looking at is, for instance, blood or semen. These are what we refer to as presumptive tests. They're not confirming for blood, and they're certainly not confirming for human blood, but when we get a positive reaction, we know that we're probably looking at a blood stain, and we need to take it a little further. If we get a negative reaction, then it's not blood, and we're not going any further with the analysis. And once you perform a preliminary test and get a presumptive test, shall we say, for blood, then what would you do? What would be the next step? If I got a positive reaction, then I would sample the stain and retain that for further DNA testing. In this particular case, when I got a positive preliminary test, then I sampled a portion of each one of the stains that I looked at on a cotton swab. I moistened that with some sterile water, and then I basically just swabbed the stain and removed it, and that was retained for further DNA testing. Just give me one moment here, Ms. Culhane. Ms. Culhane, Mr. Kratz is going to hand you a number of envelopes, and each one has been marked as an exhibit, and I would like you to look at exhibit number 155. And could you identify that for the jurors? Yes, I can. And what is that? This is a swab that was taken. My item designation was A6, and this was a swab that was taken from the RAV4. And I would like you at this point, would you look at exhibit 142? That would be the photograph, okay? Yes. And can you show the jurors on this big screen where it is that you collected this item A6? A6 was collected from the front seat driver's side portion of the vehicle. And it was a stain that I cut out of that area. And that was a blood stain, correct? Yes. You had done your preliminary test on that? Yes. I would like you now to look at exhibit 156, and could you explain to the jurors what that is? This is my item designation A7. This was collected. These were some reddish-brown crust material that was collected from this area right here on the floor by the console. And again, that appeared to be a blood stain? Yes. And what was the purpose of collecting that? I did my preliminary test and eventually developed a DNA profile from that. Now, you state that you give these an item designation number. Explain that a little bit to that. What you mean by your crime lab designation number? Okay, the vehicle itself was given the item designation of A, and everything that we subsequently collected from the car was numbered 1 through however many samples we took. So this would be referred to as A7, A telling me that it came from the vehicle, and A7 telling me exactly where I recovered this item from. And I would ask you to look at exhibit 157 and identify that for the jurors. This is my item designation A7, and this was a reddish-brown stain that was collected from the right of the ignition area in the vehicle. Okay, do you have exhibit 157? Is that A8? Yes. I thought you said, did you say A7? I didn't mean to if I did. I mean A8. Okay. Yes. So exhibit 157 is item A8. Okay, and again, what is that? That was a reddish-brown stain that was taken right here to the right of the ignition. You can see it here on the photograph. And would you look at exhibit 143, which is a photograph? Yes. And is that the photograph you have in your hand, is that the photograph that's up on the large screen now? Yes, it is. And again, before you collected this blood stain by the ignition switch, you performed a preliminary test? Yes, I did. It tested positive for blood? Yes. And then when you say collected it, just tell the jurors, how did you go about collecting it? I took a cotton swab and moistened that with some sterile water, and then I basically just swabbed the area of the stain. That material is then transferred to the cotton swab, and then when I take that back to my lab bench to do my testing on it, Then I will cut the portion that has the stained area on it and develop a DNA profile from that. I would like you now to look at Exhibit 158. That would be an envelope, and explain what that is. Yes, this is my item designation A9. And where did you take that sample from? This was taken from the front passenger seat, and this was a stain that was cut out of that area. And would you correlate that for the jurors, please, with Exhibit 144? That would be a photograph you have in front of you. Yes, this is the same photograph that's on the screen here. A9 was taken from this area right here. It was cut out of the front passenger seat. I would like you to look at another envelope, which would be Exhibit 159, and please explain to the jurors what that is and where you collected that. This is my item designation A10, and this was a reddish-brown stain that was recovered from a CD case, which was on the front passenger seat, and it's right here. And now, finally, would you look at Exhibit 160? That will be an envelope, and would you correlate that with the photographs, Exhibit 145, and tell the jurors where you got that from? This is my item designation A12, and this was also a reddish-brown stain that was found in this on this metal panel here between the back seat and the cargo area of the RAV4. It was recovered from this area right here. Now, did you perform DNA testing on each of these swabs and each of these cuttings that you've just described for the jurors? Yes, I did. And were you able to develop a DNA profile from each of those items? Yes. Now, did you also have a buccal swab from an individual by the name of Stephen Avery? Yes, I did. And once again, what is a buccal swab? That is a swabbing of the inside of the cells on the inside of your cheek. And is that referred to as a standard? Yes. And why is a standard necessary in DNA testing? Because in order to compare an evidence sample or a question sample to someone, a person, you need a reference sample. You need to know what a particular person's DNA profile is. So we use a reference standard. We know it comes from a particular person, and we can develop a profile from that and compare that to the evidence. And did you develop a DNA profile from the buccal swab of one Stephen Avery? Yes. And I'm going to show you on the PowerPoint demonstration a slide and ask if does this slide correctly display your findings of your testing of the buccal swab of Stephen Avery? Yes, it does. Would you explain to the jurors what this slide means? Earlier, when I was talking about the STR markers or genetic markers, and I told you that the PCR process amplifies or makes a lot of copies of 15 different markers, these series of numbers and letters on this side are actually designations of where those markers are found throughout the DNA. So it tells me a specific location of this marker, where it's found. These numbers on this side reflect the size of that target region of DNA that we're amplifying at that particular location. So in other words, at this location, D3, the sample from Stephen Avery, had 2 fragments of DNA size 16 and 18. And the entire DNA profile, this is what we use, these numbers and fragment sizes, that's what we use to actually compare to the evidence sample. And the example you just gave, where Mr. Avery was a 16, 18 at D3, are there other possible numbers at that location that one could be? Yes. Do you know what the range of numbers it could be? At that particular location, I believe it starts at around 11 up to maybe in the 20s. So there are numerous fragment sizes at that particular location. But here at that location, Mr. Avery happened to be a 16, 18. Yes, and at each different location, there are many choices, several choices of fragment sizes at each one of those markers. Now, did you compare this profile that you obtained from Stephen Avery's buccal swab with the DNA profiles that you developed from the blood stains in Teresa Halbach's RAV4? Yes, I did. And I'm going to put up another slide now and ask, does this slide accurately show your findings? Yes, it does. Now, in this slide, we just did, we put up for A8, A10, and A12. And again, A8 was a bloodstain found where? By the ignition. A10 on the CD case. And A12 was found on the metal panel between the rear seat and the cargo area.